As we begin the citric acid cycle, acetyl-CoA is going to be kind of our starting material that we're putting in. So in the matrix of the mitochondria, the acetyl-CoA is going to combine with this other molecule that is already there, which is called oxaloacetate. Now, oxaloacetate has 4 carbons, so when we take our 4 carbons and we combine it with 2, we end up with a molecule that has 6 carbons. That molecule is going to be what we refer to as citrate. At the same time that this happens, the coenzyme A part is going to get clipped off and that is going to leave. So we're really done with that. The next part of all of this is that citrate molecule is going to move around this cycle, and a few things are going to happen. We are going to be removing or losing carbon dioxide. We are going to oxidize the remaining food. So by remaining food, we're talking about the remaining carbons from the original glucose that we started with. And we're also going to make a little ATP. Okay, but it will just be a little. So if we look at how all of this is going to happen, every time we take that molecule and we actually see a carbon dioxide come out, we're going to see this molecule shrink a little bit. And we're also going to see at the same time a number of redox steps. And by redox steps, we mean that an electron carrier is going to come in, it's going to grab some electrons, and it's going to leave as NADH, or the reduced form of the electron carrier. So after this particular step or set of steps that I've shown you, we would have taken our 6 carbon molecule, shrunk it down to 5 carbons, again because we released a carbon dioxide here, and we've collected a few electrons in this NADH molecule. We're going to have a number of steps like this. So we will see even more NAD plus come in and grab electrons and turn into NADH. We will see even more carbon dioxide come out of this cycle. And at this point, if that happened again, we'd be left with 4 carbons there. We are then going to, and I'm just going to kind of summarize the other half here. We're going to have some ATP made. That ATP is going to be made by a substrate level phosphorylation. And it is going to be kind of a 2-step mechanism in this case. But essentially, ADP is going to gain a phosphate group to make ATP, which is a valuable product of this reaction. We will have a different electron carrier than we've talked about so far come in. This one is going to be called FAD, but FAD is the empty form, or the form that has space for electrons. So it comes in, it grabs some electrons, and it turns into what we call FADH2. And then we're going to have some more NAD plus come in, grab electrons, and leave as NADH. At that point, we will be right back where we started, in that we will have regenerated the oxaloacetate. So if we point out a few things in this whole cycle that we've been looking at here. Along the way, we said the first thing we're going to do is we're going to remove carbon dioxide. Note that right here and right here, we did just that. So we put initially 2 carbons from the original glucose into this cycle. And when we put 2 carbons in, we see 2 carbon dioxides come out. So each turn of the cycle is going to lose 2 carbon dioxide molecules. The second thing that we said is going to happen is we are going to be oxidizing the remaining food. So notice a number of redox steps. We had one here and we produced this electron carrier. Here was another one where we produced an electron carrier. And then we also had one here and we had one here. So we actually have 4 oxidation reduction reactions. And from that, if we just kind of put it over here, we do generate 3 NADHs and 1 FADH2. And again, this is per turn of the cycle. The other valuable thing that we've gotten in all of this is going to be the ATP that we made down here. So we do make 1 ATP for each turn of the cycle. With all of this said, recall that we are a lot of times talking about per glucose that we started with. So if we did want to sum all of this up and talk about what we had per glucose, remember per glucose, you actually get 2 of these. So what we would have to do is multiply all of our other stuff by 2 as well. So if we write down here what our yields are per glucose, so we are going to end up with 4 total carbon dioxides that are released. And again, these are really waste products. Those would be exhaled immediately to the atmosphere. So you'll see these just kind of go away. So they will get, again, exhaled. We are going to get a number of electron carriers that are reduced or holding on to electrons for us. We will have a total of 6 NADHs and 2 FADH2s. And then we're also going to get 2 ATPs that we make by substrate level phosphorylation. At this stage in respiration, the original carbon that we started with, so the 6 carbons that were in that organic molecule, every one of them has been lost as carbon dioxide. And that is because we lost 2 when we shipped the 2 pyruvates into the matrix of the mitochondria. And then we lose another 4 when we process both of the acetyl-CoAs that we had around this specific cycle.